In this class, we're going to look at assaying glucose and fructose levels separately using enzymes. Last time we used a DNS assay to measure the total amount of reducing sugars, not separately. Today we'll do it independently using enzymes. And next time we're going to start moving on to protein induction and purification. Glucose, which is also called dextrose, has that chemical structure. It's a primary energy source for both plants and animals. The human brain uses about 103 grams of glucose per day, so that's quite a lot. In human blood, glucose is called blood sugar. Normally, it has a concentration of about 0.1%. However, this will be higher in diabetics. Fructose is the sweetest of natural sugars. It has the same chemical formula as glucose. However, the structure is different. You can find fructose in both fruits and vegetables, though most vegetables are low in sugars. Fruits will contain fructose, glucose, and sucrose depending on the fruit type. In the United States, the main source of fructose in the diet is from high fructose corn syrup. Taking a closer look at high fructose corn syrup, it's used in many foods to lower costs, so lower quality products often use it to save money. You make it by taking cornstarch and treating it with enzymes, yielding high fructose corn syrup, meaning it has more fructose than glucose. You can vary the fructose level to get the desired sweetness, so different products might use a different high fructose corn syrup. In beverages, the syrup is typically 55% fructose, 42% glucose, and 3% unconverted oligosaccharides. Note that about 85% of high fructose corn syrup is from genetically modified plants. However, corn syrup in the grocery stores is simply glucose syrup. There's no fructose. To measure glucose and fructose separately, what we're going to do is take two enzymes from the glycolysis pathway, hexokinase and phosphoglucose isomerase, and one from the pentose phosphate pathway, glucose 6-phosphate dehydrogenase. First, we will measure glucose with hexokinase and G6B dehydrogenase. When all the glucose is used up, we'll measure fructose by adding phosphoglucose isomerase. An interesting side fact is the most common human mutation is in the G6B dehydrogenase gene, causing a decrease in the levels of G6B dehydrogenase. It affects about 400 million people, giving them increased sensitivity to free radicals. For example, fava beans and some drugs, this can lead to anemia. And often certain drugs, a doctor will check your G6B dehydrogenase to see if you have a deficiency before prescribing them. This deficiency can often lead to hyperuricemia and gout, but it does have a benefit. It gives you partial protection against malaria, which kills 2 million people per year. You will find this mutation with a decrease in activity in areas where malaria is endemic, for example, the Middle East and Africa. Note that a mutation removing function of any of the enzymes in glycolysis would be lethal. As with the DNS assay, we cannot measure glucose, so we'll measure production of NADPH at A340, 340 nanometers, just like we'll do later in the semester with NADH. Here's an example of the NAD to NADH redox reaction. Note that NADP is the same way, but it has the hydroxyl group of ribose esterified with phosphate. Otherwise, it's the same exact reaction. As with the DNS assay, we can't measure glucose, thus we're going to measure production of NADPH at 340 nanometers. If you take a look here on the graph, either oxidized NAD or NADP on the red line, at 260 nanometers, it is very similar in absorbance levels with the reduced form of NADH or NADPH, so you can't easily distinguish the two of them. However, if you move up to 340 nanometers, there is some background levels of NAD or NADP, but most of the absorbance that occurs at 340 nanometers is due to our NADH or NADPH, which is 1-to-1 stoichiometrically with our glucose and our fructose, so we can measure it indirectly using this. The first thing you need to do to your beverage samples is add hexokinase and G6P dehydrogenase. This will convert, the hexokinase itself will convert glucose into G6P and fructose into F6P. In this process, one molecule of ATP is converted to ADP. The G6P dehydrogenase can be added. It will convert G6P to 6-phosphogluconolactone, while a molecule of NADP goes to NADPH. This will cause an increase in absorbance. One molecule of NADPH will form from one molecule of glucose, so we can measure the levels that way. Our fructose, on the other hand, is sitting over on the side. It's gone to F6P, but it gets stuck. It can't go any further. So the goal here is to make sure we use up all of the glucose, measuring the change in absorbance of NADPH to give you the amount of glucose. Think about what a hexokinase does. It will take our ATP and go to ADP. It's adding a phosphate to our glucose to give G6P, whereas our dehydrogenase is taking a hydrogen and adding it onto the NADP to give you NADPH. After waiting a few minutes, we find that all our glucose and G6P will be consumed. The G6P has gone on to 6-phosphogluconolactone. Change in absorbance will stop, and that's when we know we're ready to move on to the next step. None of the F6P has been used up yet, so we have not measured any of our fructose. After the glucose, fructose, and G6P are all used up, we'll add PGI, phosphoglucoisomerase, to measure the amount of fructose. Now in this case, you can see that the PGI converts the F6P to G6P. This is a reversible reaction which will be in equilibrium. However, the G6P dehydrogenase will take the molecules of G6P that are produced and urge them to go along to 6-phosphogluconolactone until there is no more F6P left and no more G6P. It is all used up. This second change in absorbance will measure the amount of fructose. Think about what an isomerase does then, or PGI. It simply converts the two of them back and forth. Note that more NADPH has been produced from this, but this time it's only from fructose. This will allow us to separately and specifically determine the levels of both glucose and fructose in the same tube. Today, instead of using Diet Sprite like you used last week, you'll use a control solution of 0.2 milligrams per ml of each of glucose and fructose. There's no point in using enzymes on something that has no sugar. However, the control solution gives you the benefit of knowing that your reaction is working and that your calculations are correct if you calculate 0.2 milligrams per ml of glucose and 0.2 milligrams per ml of fructose. If you do not get close to 0.2 milligrams per ml for each on your worksheet, you're probably not doing your calculations correctly, so make sure you check this. Notes for today: keep all enzymes on ice and measure them carefully. There are reagents for 2 groups or 4 students in the ice buckets to share. Mix all your samples by pipetting up and down well. Don't use a pipette P20 set for a small amount. Use a larger amount, not a P1000. We don't have that much volume, but make sure you mix very well. All of your reactions are going to completion, so allow time for them to react, but it can be measured within a few minutes. Measure it, Check again, see if the absorbance has stopped changing. When it does, you're done. It also means that you can let it sit on the bench, and if you don't have a spec available, it doesn't really matter if you check it at 3 minutes or 15 minutes. The reaction will be complete. It won't go on any further. The time doesn't matter as long as you've stopped changing an absorbance. You can set up all 4 reactions at the same time to make it go quickly. And note that if you add more enzyme, your reactions have already stopped, but your levels are too low, it will not help. Adding more enzyme if the absorbance doesn't change anymore, it means you're done. You probably have not done the dilutions of your beverages correctly. Safety today, gloves and eye protection are optional.